We talked about these snail eggs, and he's going to tell you a whole lot about here in just a minute. And Andrew said this sounded like a really great project, so he kind of took part of this and got us started here. And what you're seeing today is about a quarter to a third of his thesis is getting ready to wrap up for the next semester. So, Andrew?
Thank you. All right, so apple snails are consistent of two different genera of species, but we are focused on one in particular, Palmacia. And within that, we are focused on two species, Palmacia maculata, the giant apple snail, and Palmacia pallidosa, the Florida apple snail. The Florida apple snail is the smaller one seen here, and this is actually a penny for size reference, so they're large for the snails that we're used to seeing, but much smaller than our giant apple snails, which are about the size of a palm of a hand. Florida apple snails are native to the United States, specifically around the Florida Everglades, and there are some populations up here in Alabama. Palmacia maculata, the giant apple snail, is actually native to South America, but it has invaded every continent except for Antarctica and several countries within all of those, including the United States and up here in Mobile, Alabama. Down here, I'm showing you pictures of several different eggs from different Palmacia species. The two in the red box are from the species we're interested in. The white ones here at top are from the Florida apple snail, and the pink ones down here at the bottom are from the giant apple snail. So giant apple snails are pretty harmful to the environments that they invade for several different reasons. First of all, they consume aquatic plants, and this actually led to a rice famine over in Asia during the 80s. And as we all know, what goes in must come back out, and their wastes can cause water fouling. So it can actually drop the dissolved oxygen in the environments they invade to hazardously low levels and actually suffocate other aquatic organisms. To add insult to injury, the pink coloration that I mentioned earlier in giant apple snail eggs is caused by a protein, and this protein has been shown to have neurotoxic effects in mice when injected. So naturally, since we introduced these, we thought we would try to control them, and this has proven to be very difficult. They can estivate or burrow underground during the winter to avoid cold temperatures, and they can also do this during desiccation or drying out events. They can breathe through gills underwater since they are aquatic, but as I mentioned earlier, they can drop the dissolved oxygen, and when they do that, they stick another organ called a siphon above the water, and they can use that to breathe air, so they don't suffocate themselves. They don't have very many natural predators. The only two that I've found in the literature have been raccoons, which eat the eggs, and fire ants, which also eat eggs. And we can't really use these as biological controls because fire ants are actually invasive species as well. So it doesn't make sense to use one invasive species to control another. And snail kites, which eat the Florida apple snail, typically don't eat the giant apple snail because A, their shells are thicker, and also because they're too large to swallow. And so the DNR has taken to scraping the eggs off of these substrates that they adhere to. You see that all the eggs here are laid above water. That's because if the eggs come in contact with water, they die. So the DNR scrapes them off of their substrates, and you can see all this pink coloration here in the mud from where we've done that. So our question was, does this leaching of this protein have detrimental effects on other aquatic organisms that live in these environments? In order to test this, we used a model organism in toxicology called Xenopus laevis, or the African clog frog. It's typically used in a standardized test called frog embryo teratogenesis assay Xenopus, or FETAX for short. They're used because they can be bred year-round with an injection of human chorionic gonadotropin. Their tadpoles are clear, so it makes malformation assessments really simple. And the embryos have frequently been used to predict human and environmental toxins in the past. So our objectives in these experiments were to see if and how toxic giant apple snail egg proteins were to Xenopus laevis. We wanted to see if we could alter the toxic effect by filtering this protein. And we wanted to see if we found similar effects from proteins in the Florida apple snail eggs. First thing we had to do was collect our samples, which Annie was kind enough to do for me. She went down to Mobile, Alabama, and got these giant apple snail eggs and brought them back up here. Crushed them in a Dounce homogenizer, and we dissolved them in FETAX solution, basically distilled water with some salts added back in. We removed solid particles that didn't dissolve well through centrifugation, and then we assessed the protein concentration through a Bradford assay. We had to filter some of our protein naturally for our second set of experiments. We did this through a strong cation exchange column in solid phase, and we did a Bradford assay before and afterwards to see if we lost any protein through filtration, which we really didn't. There was very little change between the two assays. Then we had to do our exposures. So we bred our frogs, and the next day they usually gave us eggs. We found them usually between blastula and gastrula stages, so fairly early in development. We removed the jelly coats from them. We put them into groups of 20 and put those groups into petri dishes. We had 4 petri dishes for controls in each experiment and 2 petri dishes for each concentration after that. And we repeated each experiment 3 times. We came in every day after 24 hours and gave them fresh solution in order to prevent that protein from breaking down. And we also removed any dead individuals that we found. After 96 hours or 4 days, we did a final death count. We assessed all individuals for malformations, and we took pictures and used a software called ImageJ to assess overall body length to see if the protein concentration affected that. Then we used a 2-way ANOVA and Cystat along with a von Fronies post hoc and probit analysis to analyze our data. So this is what one trial of one experiment looked like right here with all these petri dishes. We did experiments where we exposed African cloud frogs to unfiltered giant apple snail egg protein, apple snail protein that had been sent through the strong cation exchange column, and unfiltered Florida apple snail egg protein. These are our results. So for the next 3 slides, you'll see that we have these 2 graphs repeated with different experiments. The top one will always be mortality in red and malformation in blue, and then the bottom one will always be overall body length. The x-axis will always be concentration, and asterisks are used to indicate significant differences. So in our first results slide here, this is our exposure to unfiltered giant apple snail egg protein. The first thing that I mentioned I have on this slide is our LC50. That is the concentration at which we expect 50% of exposed individuals to die. So our LC50 for unfiltered giant apple snail egg protein is 0.55 milligrams per liter. So it's a fairly low concentration, not the lowest we've seen for any toxicant ever, but still relatively low. We weren't able to calculate an EC50 or an effective concentration for 50%, so that's the concentration where we expect 50% to have some sort of malformation. And malformations were so low that we couldn't find that for this experiment. The last thing down here is that we did have a statistical size difference here at the 0.5 milligram per liter exposure. And then we lost that here at the 1 milligram per liter. So this could potentially be due to random variation, or it could be due to the fact that we lost 90% of the individuals in that experiment due to mortality. These are the results from our filtered experiments, so a giant apple snail egg protein that was sent through the strong cation exchange column. Our LC50 here went from 0.55 to 1.88. So we basically tripled the LC50, or more than tripled the LC50 in these by simply running it through a filter. We still couldn't find an EC50, we still didn't really have very high malformation rates, but we did find a statistical size difference that was consistent after 1.5 milligrams per liter. And these were our results from the Florida apple snail egg protein that was unfiltered. So you see there's a lot less depth here. We were unable to find an LC50, EC50, or statistical size difference. And I would like to point out that on these graphs, the highest concentration that we tested were 2.5 and 3 milligrams per liter. Whereas in this one, we went all the way up to 10 milligrams per liter, where we still didn't see an LC50, whereas the other two, it would cause 100% mortality. So what we conclude from this is that Palmacia maculata, or the giant apple snail egg protein, does have a toxic effect towards Xenopus laevis, and it has an LC50 of about 0.55 milligrams per liter. The toxin has a greater effect on mortality than malformations, so it is non-teratogenic, does not cause malformations, it simply kills the individuals exposed to it. We reduced this toxicity through the strong cation exchange column by 3.7 fold, so it's a pretty drastic difference. And P. pallidosa eggs, or the Florida apple snail eggs, either do not have very much or have none at all of this protein present within them. So this toxin that we've been experimenting with is novel to the environments that these snails are invading. I want to thank the JSU Biology Department for supporting me throughout my thesis, the faculty research grant, which helped us to get a lot of the money that we needed to do this, the Kensaw brothers, who have helped me to travel to a couple different places and present this research, and Annie Slayton, who Dr. Rayburn mentioned earlier, who was the source of a lot of the pictures that you saw, the eggs that we received, along with being a walking encyclopedia for apple snails. So she's really helped me with my thesis. Those are my works cited, and if we have time for questions, I'd like to take a few. Yes. Hi.
So when you filtered the apple snail eggs through
Yes.
the filter, did that remove the pink color, the pink that would lead to the toxin that makes things sick, or
It did
what
not.
happened?
The filtered and unfiltered basically looked the same. There's always that sort of, almost kind of penny, like coppery colored pigment to it. Yeah.
So what was the purpose of the filtering, I guess?
We wanted to see if we could, we were trying to prove if it was proteinaceous in nature. Basically, we were seeing if we could sort of tear apart that protein through the interactions between the solid phase filter and the protein itself. Or we also wanted to see if we could filter that protein out, but that clearly didn't happen either, so.
Related to that, you measured the protein concentration both before and after
Yes.
filtration. What was the difference?
There was no difference, or there was very little difference, if any. Anybody else?
All right, here's the green.